Okay, so we're going to very quickly make our way through the kidneys, and we're going to talk about some of the functions of the kidneys starting out. This is going to be in two parts. So the first part is just going to be getting to be familiar with the kidneys and the nephron, which is the functional unit. So we'll talk about the function, and really that's the regulation of extracellular fluid volume and blood pressure. So how much fluid is in your body is directly related to blood pressure. As we've mentioned before, if you put more fluid into a container that's only so big, then that's going to increase the pressure within that. And in this case, we're the container. And the more volume we have, the higher the blood pressure. That's one very simple way to look at it. And so the kidneys have the power to excrete anything else. If they don't want as much fluid, they can excrete that as urine, and that will lower the fluid volume. So that includes the extracellular fluid volume, and then of course how that relates to blood pressure. So that means that they work kind of in concert with the cardiovascular system, and a lot of blood pressure drugs that we have are actually drugs that work on the kidney. They're things like diuretics, and a diuretic is something that makes you urinate more. So that obviously would make sense to lower volume, fluid volume in the body, and therefore blood pressure. Now, regulation of osmolarity. Now, our cells like to be at a particular concentration. The magic number is 300 milliosmoles per liter, or milliosmolar. That's about where we want to keep things. So when we get dehydrated, this tends to start to creep up. And if we are overhydrated, this tends to creep down, and so we'll start to collect and preserve sodium and things like that. So we're trying to keep it kind of in this range within our cells, between our cells, and of course what's outside our cells, the concentration is going to be the same in both sides. One of the main things that affects osmolarity is sodium. Okay, so about 90 to 95% of our osmolarity is actually controlled by sodium. So you can imagine that the kidneys play a big role in making sure that we have the proper concentration or the proper amounts of sodium in our body. If we have too much, it will be excreted as urine, and if we don't have enough, it will be reabsorbed back into the body, as the kidneys will get that sodium, and then they'll say, wait, the body needs that, and then it will be reabsorbed back into the blood, so the body can use it to maintain fluid volume, to maintain osmolarity, and all of that good stuff. Now, another thing that happens is the regulation of pH, and it's really pretty simple the way the kidneys regulate pH. If you have a low pH, or if you have acidosis, then the kidneys will simply get rid of that. They'll simply get rid of it. We also know that buffers and respiration can control pH, but the way the kidneys do it is pretty direct. If your pH is too high or you have alkalosis, then again, your kidneys will just get rid of this bicarbonate, and then that will be excreted as urine, and that will adjust your pH. It's a little slower than the buffer, of course. The buffer is pretty immediate, and then respiration is fairly quick too. This is a little slower, but it's very direct. Okay, um, so that's an important thing that the kidneys do. And then excretion of waste. Obviously, your blood, because really what your kidneys are doing is filtering the blood. And so all of these wastes will show up in the blood, drugs will show up in the blood, and other toxins will show up in the blood. And so the kidneys, the role of the kidneys is to filter the blood. Okay, so they're constantly um, filtering blood. Okay, and so any of these things will then again be excreted as urine. Okay. And then the production of hormones. Now we're going to talk about that in this next section, but that's another thing that happens in the kidneys is hormonal control, hormonal production. Some hormones, obviously not all of them. Hormones are created all over the body, but the kidneys make some, and most of those are involved in something having to do with the cardiovascular system, blood pressure, something like that. Okay, so to get a good idea of the anatomy of the urinary system, most of what we're going to be talking about is what happens inside the kidney, but it helps to kind of have this backed out view so we can actually see what's going on. So we have the heart up here, and then there's the descending aorta and the abdominal aorta, and so blood goes there, and then it kind of shunts over here, and about 20 to 25% of your cardiac output will actually move into the kidneys. And then what isn't reabsorbed back into the body is then excreted through this ureter. So there's this peristaltic movement. The kidneys are constantly producing urine, and so it moves through this ureter, and then it's stored in the little in the urinary bladder for a little while, and it will expand and get larger and larger as it fills with urine. And then we have the micturition response, which is where urine will move out of the urethra and then to the outside world. Okay, so that's really all we're going to see of, we're just going to get closer and closer into the internal workings of the kidney, and we're going to leave all of this other stuff behind. But if we look a little closer, we can see that here's the kidney. This is the renal artery, so this is where blood is actually entering, and then that will divide into other smaller arteries, and then arterioles up here that will actually feed into something called the nephron. So if we look at this, it's just like everything else. The outer layer is the cortex, the inner layer is the medulla. The medulla starts around, these guys are called renal pyramids because they look like pyramids. Okay, so you have a whole bunch of those. And the medulla actually starts at the outer edge of these pyramids. Okay, let me maybe use a different color here. So at the outer edge of these pyramids is where the medulla starts. Okay, and that's kind of important because the concentration deep in the kidney is very high. So it has high osmolarity, so which is just high concentration. On the medulla side, so on the inside, and then out here it's a little more dilute. Okay, and that's a pretty important concept when we get to the next section, that where it's supposed to be dilute. When we get to the next section, when we actually start talking about how you either, because we know this, we either have very concentrated urine or we have very dilute urine. And it's that concentration gradient that does that. So yeah, cortex, medulla, because we're going to have these things called juxtamedullary tubules and things like that, and so it helps to know that we're talking about the medulla when we say that. Okay, and then I mentioned the pyramids, and then these arteries that are dividing will divide, and then they form something called afferent arterioles. That's going to make more sense when we actually look at those. But those afferent arterioles are going to feed into something called the nephron, and then that's going to dip down into this more concentrated area, and that's how urine's going to be made. And remember, I said kidneys get like 20 to 25%, a fourth of all the blood that's being pumped goes to the kidney. And all of that blood that goes into the kidney or into the nephrons, these guys, is on its way to being urine unless it's reabsorbed. So we're going to talk a lot about that. All right. So blood and nerve supply. As I mentioned, although the kidneys are 0.5% of the total body mass, they get 20 to 25% of the resting cardiac output. Left and right renal artery enters, branches into arteries and arterioles, and then each nephron, and this is the start of a nephron, this thing would go on, this is the proximal tubule, and then the descending loop of Henle, and then there, if we were to keep going, there would be an ascending loop and a distal tubule. Okay, but we're talking mainly about blood flow here. So each nephron receives one afferent arteriole, which is what this guy is. Okay, so this is the afferent arteriole right here. Okay, and so blood flows into that, and then it goes into something called a glomerulus, where it's filtered, and then it enters that nephron. So anything that moves into this area is on its way to becoming urine, except there's this thing called reabsorption that we'll talk about. Okay, so it divides into this big, well, not big, but a capillary ball. Okay, so that's what this is. This is a little capillary ball, and we call that a glomerulus. Okay, so that's what this thing is here. This is the glomerulus. Okay, now something very unusual happens in the kidneys, and that's on tests, and it's something that people like to talk about. But you have an afferent. Afferent means it's going to. You have an afferent arteriole. Okay. Let's see, afferent arteriole here, which I had marked, and then it goes to a capillary. This is a capillary ball. Okay, so this glomerulus is a network of capillaries, and then it leaves to form another arteriole. Now that's unusual because we're used to going arterial, capillary, and then venule, but in this case we're not losing any oxygen. It's just being filtered, and so this is the way it goes there. Okay, so afferent, afferent arterial, capillary, and then efferent arterial. Okay. Okay, so let's see. So let's look at where we're going from there. And you can see it kind of here, this is the afferent arteriole moving in. So we're up here, afferent arteriole moving in, and then there's the glomerulus, and then you have this thing called filtration, and then the efferent here. And so it feeds into this nephron. Okay, so we have a couple of different types of nephrons. There's the cortical nephron, and then there's something called the juxtamedullary nephron, and it juxtaposes down here to the very concentrated areas and then back up. That's the loop of Henle, this part of it, okay? To the distal tubule and then to the collecting duct. So once filtrate, so filtered blood, makes it into here, then it's ultimately going to move down here, and then that's going to go to the ureter that we talked about, to the bladder, and then to the urethra to be excreted as urine. Okay, so this is the nephron. It's the microscopic functional unit of the kidney. We have like 1 million of these. Okay, so that's a lot. And that's really what the kidney is, and that's what the filtration is. That's where it takes place. Okay, so we're going to spend a lot of time talking about that. Okay, so let's look a little more closely. Afferent arteriole, and let's talk about some of the parts of it. Okay, so if we take this and we kind of unfold it so it makes a little more sense, really it loops around this way because this descending loop, or yeah, the ascending loop actually goes right past the glomerulus here through this, between this afferent and efferent arteriole. Okay, but if we unravel it, it actually makes a little more sense. It's easier to understand. So blood moves in here, and then it's filtered, and then it moves into this area, and this is called the proximal tubule. It's called proximal because it's close to this thing called the Bowman's capsule. Okay, so the Bowman's capsule is just this outer covering that surrounds the glomerulus, and the glomerulus is this little thing in here. Okay, so together that's called the renal corpuscle. I won't ask that on a test, but this entire thing is called the renal corpuscle, and then that's broken down into the Bowman's capsule, which is the outer edge, and then the inner part, which is the glomerulus. Okay, so this Bowman's capsule will actually continue to make the proximal tubule, and then you have the loop of Henle. Okay, so that's what it's called, and that's how you pronounce it, Henle. Okay, and you have the descending limb, or the descending loop of Henle, and you have the ascending limb, the ascending loop of Henle. Okay, and then you have the distal tubule. If this was proximal to the Bowman's capsule, then this is distal. Okay, so this is the distal tubule, and then you have finally the collecting duct, and the collecting duct will then drain, and that's what actually becomes urine. Okay. So if a fluid or some particle makes it through here all the way to the end without being reabsorbed, then it becomes urine. Okay, so let's look at how that happens. So I keep talking about how things go in to the afferent arteriole, and then they get filtered into the nephron, and then they can be taken back up into the blood. And see, it's because we have this blood supply all the way around this, and we call these paratubular, para means around or next to, paratubular capillaries. And so anything that we want to keep, let's say that we have glucose, and glucose will make it into the nephron, but we don't want it to go all the way through. We don't want it to turn into urine. Okay, so what happens is it can be reabsorbed into all of these blood vessels that are sitting around here, and that actually mostly takes place in the proximal tubule. We'll talk about that. Okay, so these blood vessels and the close association of these capillaries, paratubular capillaries, is very, very important. Now, there's another thing that we're going to talk about in the next section that's going to involve these, which are other types of capillaries that sit around the nephrons. They're primary in the juxtamedullary, the ones that are really long that go all the way down in the medulla. We have these things called vasa recta, and those help play a role in getting very, very concentrated urine and forming that concentration gradient, which causes very high, because remember we said in the blood cells, in the blood, it's usually about 300 milliosmoles. You don't necessarily have to know what that means, but that's just a concentration, and the bigger that number, the more concentrated it is. So down deep here, it's about 1200. Up here, it's about 300. So as it moves out of the Bowman's capsule, really it's about the same concentration. It's about the same as blood plasma. But then as it goes deeper and deeper, it gets more and more concentrated, and it sort of squeezes all that water out, and then you have some fine tuning that takes place up here, and that's what the next section will talk about. Okay, so I keep talking about filtration, reabsorption. I haven't mentioned secretion yet, but that also takes place. So filtration is the first thing that's going to take place. Okay, so filtration is just this process by which the blood sort of flows through here, and then large things like blood cells, if you can imagine these as being blood cells that are flowing through there. I don't know how to scale that is, but these blood cells, those will just keep going. Okay, so you don't want to get rid of blood cells. You don't want blood cells, red blood cells or white blood cells, in your urine. You want those to keep going. So that gets filtered out. And so the only thing that really goes through here is, I'm going to put this, and I'm going to put it in quotation marks, plasma. Okay, remember plasma is the liquid part of your blood. So really in the Bowman's capsule right here, all you have is plasma. Okay, it's blood plasma because it's everything except large proteins and blood cells. Okay, so that's what the filtrate is immediately. And then things start being reabsorbed. Okay, so that means that things like glucose, sodium, chloride, potassium, urea, which is a breakdown product of proteins, all of those things can be reabsorbed. And there's also secretion that takes place. So all of these things, we want most of that stuff. Water is another one. Water is very important, right? We would 25% of our blood volume going in here. If we didn't reabsorb that, we would dehydrate within a few minutes. So we also have secretion that we'll talk about. And secretion is just things that maybe there's a toxin or something that most of it got in here, okay? But some of it got passed, and so that can actually be secreted, and more of it can be added. And we'll see an example of that. Penicillin is a good example of that. So when we put this all together, so if we can imagine sort of taking this is the glomerulus, this is where filtration takes place. If we take all of these things together and we assume, okay, well, a certain amount of a material has moved into here, but then some of it has been reabsorbed, okay, and maybe some of it has been secreted again. Okay, so it's all over the place. Some of it has been secreted. Well, what ends up going out is what we call the excretion, or what's excreted. Okay, so the amount filtered minus the amount reabsorbed, because that leaves, right? Minus, it's negative, it's going away, plus this is being added, the amount that's secreted is going to equal what is excreted as urine. Okay, so filtration is the movement of fluid, or the movement of blood into the nephron. And when we say blood, we really mean kind of more like blood plasma. Okay, so the fluid part of the blood. And that's going to take place in the glomerular capillaries in the Bowman's capsule here. Okay, and then reabsorption is moving substances from the lumen of the nephron back into the blood. That's absorption. The blood absorption is just when something moves into the blood. Secretion is when something moves out of the blood. Okay, so secretion moves selected molecules from the blood and adds them to the nephron tubule. Okay, so let's start with, we're going to go through each one of these things individually. Let's start with secretion, or filtration. So filtration happens. Here's our afferent arteriole that's leading into the nephron, and this is our glomerulus. And so the blood moves in and moves all through here. and you've got a certain amount of pressure pushing the blood through here. And so all we need to do is open little pores or slits, and they're actually called fenestrae. Um, so our fenestrations, which are just pores. Okay, so these are little pores. So you can imagine a large blood cell being in here, and that would not be able to go in. So that would just keep going. So if we go back over here, the blood cells will just keep going. Large proteins, so we might imagine like a large protein here or something, and those might not fit in. So they would continue to the efferent arteriole. However, small things, little things like sodium, chloride, glucose, the smaller ions, electrolytes, molecules like glucose, urea. Urea is small. That's basically just what ammonia. So your proteins will form ammonia, the amino acids will form ammonia, and then they form urea, and the urea can move through here. So all of these things can move through. So what you have here is basically plasma. Okay, I hope that makes sense. Okay, because it's in the blood here, and then as it's filtered out, you end up with just plasma on this side. Okay. It's just missing a few proteins and blood cells, which is kind of what plasma is. Okay, so let's talk about this. We call this, because it happens in the glomerulus, we call this glomerular filtration. And it works just like, if we go back here, it works just like a spaghetti strainer almost. I mean, you can imagine that, you know, a spaghetti strainer, you put the spaghetti in, and then the water drips through, but the spaghetti stays up there, okay? So in one way, that's how it works. In another way, we have a lot of pressures that are pushing against it. And the main pressure, I'm just going to say this, we do have some backflow. So there's pressure pushing back because you can't fit, because you're trying to push fluid in here and there's only so much space. So that's kind of pushing back. And we also have, because we have all these blood cells, we also have some osmotic pressure pushing back. But overwhelmingly, we have this thing called hydrostatic pressure, which pushes it in this direction. So we always have, unless you have a malfunction or some kind of kidney disease, you always have fluid being pushed into the nephron. Okay, and in fact, it's about 150 to 180 liters, but of all of that fluid that's moving in, most of it is reabsorbed. Okay, so filtration of water in small solutes happens at the glomerular filtration, prevents the filtration of most plasma proteins, most of these larger proteins, blood cells, and platelets. I didn't mention platelets, but that's part of that group of blood cells. Okay, so volume of fluid filtered is large because of large surface area, and that's what this is doing. It's trying to get the biggest surface area possible, so we have filtration that's happening all over the place, and this fluid will then all move into the proximal tubule. Okay, so it's a large surface area, thin and porous membrane, and high glomerular capillary blood pressure, and that's what I was saying. That's that blood pressure that's pushing in this direction. Okay, so that's how we keep blood moving through there, and technically, like I said, it's plasma moving through there. But we want that to happen at a consistent rate. Because as things are being filtered, you can imagine that if the rate was very, very fast, okay, remember a lot of things have to be reabsorbed. So if it's happening very quickly, well, that stuff's not going to get reabsorbed very well. If it happens too slowly, okay, so if it's a very slow process, then maybe we absorb too much. It's being reabsorbed all the way along there, and maybe we're going to reabsorb more than we need, and maybe some toxins will get back into the body. So we want that to happen at just the right rate. And so as our blood pressure increases, that's what this graph shows. It shows that as your blood pressure increases, your mean arterial pressure increases, your glomerular filtration rate, the rate at which that fluid is being filtered, is really, really constant. Okay, up to a point. Up to a point, it just can't hold it back. The pressure's too high, and then here it's just too low. You're not getting enough blood in there. And so that has to be controlled. We have to have a way of controlling our glomerular filtration rate. And guess what? That's done through the afferent and the efferent arteriole. They have smooth muscle. They can let more in, or they can keep more, or they can keep some from coming in. Okay, so glomerular filtration rate, that's the amount of filtrate formed in all the renal corpuscles being the glomerulus and the Bowman's capsule together, of both kidneys each minute. Okay, so that's what our GFR is, and you're going to hear a lot about GFR. Okay, so if it's too high, substances pass too quickly, so I mentioned that. And if it's too low, everything or too much may be reabsorbed, and we may actually keep some toxins or some waste products that we wanted to get rid of as urine. Okay, so GFR is directly related to the pressures that determine the net filtration pressure. So let's talk about how that happens. So the way that the kidney or the nephron is made is you have this proximal tubule, descending loop of Henle, and then it goes up here just where it starts to become the distal tubule. So right here in this area, it actually goes right past the afferent and the efferent tubule. So all your kidney has to do is you have this group of cells, they're called macula densa cells that sit right here. All they're doing is they're constantly measuring how much salt is in here. They're measuring the blood pressure. Okay, so they're measuring all of this stuff, and then they will pass that signal on to the efferent and to the afferent arterioles. And that will adjust if, let's say, the pressure is too high. It might say, hey, the pressure's way too high. Things are moving through here too quickly. So this is supposed to be the distal tubule. It's cut away. So that's this area here. And so it'll say, hey, things are moving through here too quickly. I think we need to slow it down. So then this can constrict. Okay, so it'll constrict to make that hole a little smaller, which slows blood flow, which is going to slow your GFR, your glomerular filtration rate. Okay, so myogenic response, that means muscle, and it's autoregulation. It's taking care of itself for the most part. Now, that can be overridden by hormones and autonomic nervous system. So this if you go into shock or something like that, then that may actually cause contraction of this. So if you're in shock, your blood pressure is falling, you're bleeding somewhere, well, one of those responses might be to just shut this off. That's not a time we need blood flowing through here. That's a time where we need the blood to go right past there and just stay in the body. Okay, so that increases the blood pressure out here. Okay, so there are lots of ways to control it. It's either autoregulation, where it will then have that paracrine control to say, hey, we need to constrict or we need to dilate, and you've got this constant, it's constantly constricting, dilating. So these muscle cells are either opening up more or they're closing down, and that's trying to keep your glomerular filtration rate steady. Okay. So that's the purpose of that. And it's monitoring it through this thing called the Juxtaglomerular Apparatus. And that's what this entire thing is here. Okay, so let's say we want to change our GFR. Let's say things are moving through too quickly. So we have, it's very, very fast moving through there. And we want to slow it down. We want to decrease that a little bit. So what we do is we tighten or constrict those afferent arterial muscles, okay, the smooth muscle surrounding them, and this is showing it as a clamp, okay, and we close that off a little bit, and that will lead to a decreased GFR. Now, another thing we could do is instead of closing these, we could open them, and that would increase GFR, okay, or we could close these down, okay, up here, because you have the same types of muscle situation in the efferent. We could close that down, which is going to slow the blood from moving this way, which is going to push more out. So by adjusting the afferent, most of it happens in the afferent, okay? So most GFR regulation occurs at the afferent, but some also occurs at the efferent. And it's really just getting that signal saying either to constrict or dilate, either let more through or less through, that will then affect how quickly things are filtered, and that's your GFR. Okay. All right. So let's see. Trying to figure out where we were. Okay. All right. So once we've moved into, so we've had our filtration, we've moved through. Okay, and so basically right at this point we have blood plasma. Well, now we have to get stuff back, right? Because we don't want, first of all, most or all of your glucose has got to be reabsorbed. Your sodium has to be reabsorbed. All of this stuff has to go back into the bloodstream. I will say, first of all, that sodium is actively pumped out. Okay? When I say pumped out, I mean pumped out of the filtrate, pumped out of the nephron, reabsorbed back into the blood. So it's actively reabsorbed. And by actively, I mean the way that we think about active in physiology, which is it burns ATP. Okay? And what better way to pump sodium than with the sodium potassium pump? Okay? So the sodium potassium pump really gets things going. Now, this is where we have to think a little bit. If the sodium leaves, what tends to follow the sodium? So the sodium is actively pumped out. What follows sodium? Usually chloride. Okay? Chloride, there might be some phosphate ions too, but mainly when we talk about anions, we talk about chloride. So now we have, instead of salt, this is the filtrate, okay? So this is inside the tubule. So now instead of having the salt in here, the salt is now out here. So the sodium is out there, the chloride is out there. If we remember osmosis, then what happens next? Well, the water says, hey, what happened to all the salts that were in here? Okay, I wanted salts in here, but now all these salts are gone, and so water naturally will try to even out that concentration. It'll say, wow, it's really concentrated over here. All of these salts have moved over here. So what is water going to do? Water's going to leave. Okay. Remember, this is all happening right now in the proximal tubule. And so the water will move out. And then we have this higher concentration of potassium, calcium, urea. All of those things are now at a higher concentration, so those will move out. So think about this. All you have to do, all the kidneys really have to do is pump sodium out. Sodium is reabsorbed back into the blood, out of the nephron by active transport, the sodium-potassium pump. It's pumped out. And then you have everything else that just sort of follows it. The chloride will follow it. Once those salts leave, the water will move through osmosis. Okay, that's how the water moves. And then the potassium, the calcium, and the urea will all follow suit. They'll all say, well, it's getting very, very concentrated in here. My concentrations are building up. Everything is leaving, and so they will then also leave. Okay. All right, so there's one thing that I want to talk specifically about, and that is glucose, okay? So glucose, it's very important because of things like diabetes, and we know that if there's glucose in your urine, something is wrong. You shouldn't have glucose in your urine. And so glucose is moved out by this co-transport. So as sodium is being pumped out, there's our ATP, that's our sodium potassium pump. So sodium's constantly being pumped out. And then it's empty in the cell, so all of the sodium that's over here, all you have to do is open up a channel, and the sodium's going to move in here, and then it's quickly whisked to the other side. Okay, so all you have to do then is just say, well, sodium, that concentration gradient of sodium, is going to pull glucose, and that's a glucose transporter. Okay, and so we have a bunch of these all along the proximal tubule, and they're all trying to pump glucose back here, and then once the glucose gets pumped in here, then the glucose will leave the other side, okay? Because it gets built up in here, all these glucose molecules, and then there's a concentration gradient, and then it moves to the other side, okay? Now, just imagine if we had so much glucose in our renal tubule, in this tubule over here, we have so much that these are all clogged up, okay? So they become saturated, and we have glucose just waiting. Okay, so if your insulin isn't working, your insulin isn't getting rid of the glucose, then you have a big line of glucose that's waiting to get through. That has a lot of implications because if you're not able to get rid of all of your glucose, then the glucose stays in here. Now remember what water wants to do? Water wants to go to where the concentrations are highest. Okay? Well, if the glucose builds up in here, guess what's not going to leave as much? Water. It's going to say, oh, actually the concentration's really high in here. So I'm going to stay in here too. So if we think of the blue as water, the blue has to also stay in here to kind of even out that concentration. Since the only way that this does not become urine is if it's reabsorbed back into the body, then this water stays in the tubule, and then ultimately it will be excreted as urine. Okay, so having higher glucose saturates these glucose transporters, so the glucose can't move out. The glucose stays in the tubule, which keeps water in the tubule, and you have increased urine. Okay, so you have glucose in your urine, you have increased urination. That's a lot of times one of the symptoms that people will have is they're urinating all the time, high volumes of urine, and then they go to the doctor, and the doctor measures their glucose concentration, and they find glucose in the urine as well, and that's when usually that's a diagnosis of type 2 later in life, but type 1 would have the same effect when you can't control the glucose levels. Okay, all right, so one thing we haven't talked about yet, the last thing besides excretion, is secretion. Now we see that there's reabsorption that takes place, and most reabsorption takes place in the proximal tubule. But something else takes place, something else takes place all the way along. And when I say all the way along, it's mainly in the proximal tubule and the distal tubule, and then a little bit in the collecting duct, is secretion. So that's this idea that if something made its way past and didn't get completely, didn't make it through the filtration process, okay, so this is what I mentioned earlier, and I have another picture that'll show that it will actually move back into the tubules through this process called secretion. Now, a couple of things that your body wants, and it happens all the time, is potassium and hydrogen. Okay, depending on the pH, depending on your potassium levels, you're going to have potassium that's secreted back into the tubule, and then it will be excreted as urine. Okay, I'm going to show an example in a second about how penicillin works. Okay, so remember that we have filtration. First of all, what leaves, so we subtract out what's reabsorbed, and then we add back to that what is secreted. Okay, whatever makes it out of the collecting duct, this part is the collecting duct, okay, whatever makes it out of there is excreted, and that's urine. Okay, so excretion equals filtration minus reabsorption plus secretion, and we call this clearance. So when we have a toxin, or usually when we talk about this, honestly, we're talking about drugs, clearance of drugs. Okay, so how quickly that drug can be cleared in the urine. Okay, and drugs have different clearance rates, and so if it's secreted and not reabsorbed, then it's going to be a much faster clearance, okay, because none of it's leaving. Okay? It's all staying in there. Plus, if it's also secreted, you've got even more being added, and that's an example of penicillin. Now, inulin is one they test for glomerular filtration rate, and all they do with that, that's neither secreted nor reabsorbed. So what's in your blood, the concentration that's in your blood, should be the concentration that's in your filtrate, or at least the amount that's moving through. Okay, should be roughly the same. So you can actually measure that to figure out what your glomerular filtration rate is. However, this is penicillin, and this used to be a problem back in World War II when they first discovered penicillin, started using it, that the clearance for penicillin was very high. So let's just look at something. We have about 100 milliliters per minute being filtered. Okay, you see that? 100 milliliters per minute being filtered. That's how much penicillin is moving through. If we look down here at how much is being cleared through and we've left off a lot of pieces here, but if we could imagine this as being the collecting duct, and we've gone through the proximal tubule and the loop of Henle and all that other stuff, the amount that's actually excreted is 150. Okay. Now that doesn't make any sense, right? Because if 100 milliliters per minute is moving into the tubule, and then 150 milliliters per minute is actually being excreted, where does the rest of it come from? Well, the rest of it comes from secretion. Okay, so that means that some of this penicillin up here is filtered, and that's fine. There it is. But some of it makes it past. Okay, so some of it didn't get filtered. Well, guess what? That stuff that didn't get filtered, there's a pump. Okay, it's a general pump. It's an organic molecule pump, but that will then pump that penicillin back into the nephron, and that is secretion. Okay. So when our numbers don't add up, it's because either we've lost something to reabsorption, which is usually the case, or in this case, we've actually gained some through this secretion. Okay, so hopefully that makes sense. Now, the next thing we're going to talk about in the next section is how this stuff is controlled. So we're going to start talking about hormones and things like that that will actually change how you have reabsorption or filtration. And most of that will take place in the distal tubule. So what we've been talking about, distal tubule and collecting duct. Okay, so what we've been talking about is happening all the way along, primarily, like I said, in the proximal tubule. But it's also happening along the descending loop, which mainly we have just water moving out in the descending loop, and then the ascending loop, which mainly we just have salts moving out, okay, being reabsorbed. When I say moving out, that means reabsorbed. But then the control that we're going to be talking about next is actually in this area, and things like antidiuretic hormone or vasopressin, aldosterone, hormones like that have a major effect on how concentrated the urine is. Okay, so urine concentration. Because at the point here, at the beginning of the distal tubule, all of this stuff has happened pretty normally. The salts leave on their way down. You can see that the descending loop of Henle gets a little bit thinner because of the salt, or the water, I'm sorry. The water leaves, and the descending loop of Henle gets a little thinner because the water is leaving. And then here the salts start to leave, and it becomes more dilute, and the water kind of collects in here and starts to get larger again because the water is just kind of collecting, the salts are leaving. And so you can kind of adjust that volume. But at this point, it's almost always about the same, okay, for everyone and for every nephron, okay, the concentration. Okay, so concentration is about the same. Concentration, okay, and that's because the reabsorption and secretion has happened pretty normally. Okay, so now we have a lot of control because we can either get it very concentrated, which is 1200, remember that's what it is deep, or we pump out so many salts in this area in the ascending loop of Henle, this actually gets down to about 100. Okay, remember 300 is where most cells in your blood are. So we have that variability. So it can be, your urine can either be very dilute at about 100 or it can be very concentrated at 1200, and that control takes place right here, which is what we'll be talking about in the next section.